Everyone, it's Dr. Paul Rose again, and in this presentation we're going to look at the role of natural history specimens to wildlife conservation. What can we learn about the specimens that we have in museums that tell us information that's relevant to conservation action? And how does the information held within museum specimens allow us to really get under the skin, bad pun, of wildlife conservation challenges? Natural history museums around the world are full of priceless specimens that tell us about current extant biodiversity as well as extinct biodiversity, and we can use that information in conservation planning and conservation action, and we have a better understanding of our impact on the natural world by looking at all of the specimens of extant and extinct animals in museums so that we can face and meet the conservation challenges of the future by looking at some key aspects of the past. Museums convey the diversity of life on Earth at an extreme scale. They can show us all of the different forms and variations that animal species and subspecies come in, like this ungulate display at the Natural History Museum in Tring. It provides a snapshot of how evolution has caused animals to adapt to particular situations in a particular way. By looking at these specimens and understanding their form and function, we are able to understand the evolutionary processes that caused species to change in this way. And therefore, that information is invaluable to how we try and conserve living specimens to allow them to further adapt and evolve into the future. Because that's what conservation of the living animal is all about. It's conservation of the genes within that population that allows for future adaptation. So by understanding where animals have come from, by comparing species and by comparing populations, we can see how evolutionary forces have acted on these particular individuals and how that shaped the evolutionary path down which they've trodden. So I'm going to take a walk virtually to the corridors of the natural history museums of the world, looking at some of the specimens they contain and thinking about the value of these specimens to wild world conservation. What can we learn from them? How does studying the animals they contain allow us to move forward with the decisions that we take that influence conservation action in the zoo and in the field? Museums are obviously full of biological specimens, and that means they're great places to study speciation. Let's look at the anseriforms, the ducks, geese and swans. Here we've got a case full of different species of ducks, shelled geese and various goose-type ancestors. We can see how some species keep similar features to others, but how other species are completely different and have evolved traits that we don't see in near relatives. Yet, there are features of all of these birds that suggest they are closely related. So direct comparison between individual species within a taxonomic group is possible in displays like this. We can look at conserved features and we can look at the adaptations and evolutionary pathways of brand new features that individual species have evolved. And from a conservation perspective, we can have a look at how and why some features have been conserved across the evolutionary timeline of a species and why that might be important for how they fit into their habitat. And even within a taxonomic group, where individuals might look incredibly similar, we can look at specific differences between species that create just that. It's evolved into particular species of animal. These subtle differences cause individual communities of animals to breed together, which creates the species overall. In this example here, we've got the Berkshire zebra, the plain zebra, the mountain zebra, and some of the wild ass ancestors of the zebra too. Different stripe patterns, stripe thickness and stripe length denote the different species of zebra, and therefore where the zebra has come from, why the zebra has evolved, and how the different individual species of zebra fit together in the zebra family tree is possible thanks to these museum collections. And if we need to study where the zebra might be going from an evolutionary perspective in the wild, we can look back at where the zebra has come from and all of that information contained within these museum specimens to help us understand the evolutionary pathway that that species is on. And that's particularly important for extinct species or extinct subspecies of animals. This is the Berkshire Zebra, which is an extinct subspecies of zebra. And you can see in the main photograph that this animal had very pale stripes towards its rear end. It's very similar to the Quagga, which is the extinct species of zebra from South Africa. In fact, the Quagga gives its name to the whole complex of plains zebra species and subspecies, Equus quagga, and all of the subspecies are denoted from that. So clearly there is a pattern and a geography to how zebras get their stripes. In South Africa, they're less striped, and as you move up the continent, as you travel north, so zebra subspecies of the plains zebra complex get more striped. We can look at the reasons for this. We can look at how these different subspecies of zebra fit together, both biologically and geographically, by looking at museum specimens. And we can also take genetic samples from these museum specimens, such as this Berkshire zebra, that might tell us about the genome and the genomic differences between these different subspecies and how that might inform future conservation action. How could we potentially try and replace some aspects of lost biodiversity by conserving the adaptive potential, the patterns, colour morphs and subspecies, and all of that information that's encapsulated in the current living, breeding and evolving individuals in wild populations that are still around. Here is a specimen of the quagga from the Natural History Museum at Tring in the United Kingdom. You can see it looks very similar to the Berkshire zebra. Yes, it's much browner, but it also loses its stripes as you progress down its body. The head shape is very similar to the Berkshire zebra. So we've got this nice example of how species and subspecies might look similar, but subtle differences in their location cause them to be distinct enough to be separately breeding populations. There are projects set up in South Africa to try and bring back the quagga, at least in appearance, by looking at these southern populations of zebra subspecies that are naturally less striped and are breeding them to reduce the striping that they have on their back legs and rump, and therefore they resemble the quagga. Museums have some very famous examples of extinction events preserved within them. Preservation is not the same as conservation. Preservation is like jam. You're preserving that fruit in some form. These great auks, an extinct flightless seabird, are preserved. There's no adaptive potential to these specimens. They're preserved in the form that they once lived. Conservation is all about conserving that species and the chances that that species can continue to breed, evolve and adapt into the future. The great auk went extinct in 1844, the last two birds actually killed for museum collections. A sighting of a live bird in 1852 has been accepted by the IUCN as the last documented report of the species being seen alive. In the middle photograph of the great auk with the egg, you will note that there are some jars behind this bird, and these jars contain some very interesting information. In the Natural History Museum in Copenhagen in Denmark, are the stomach, eyes, and various other internal organs of one of those last two great auks that was killed in 1844. These are clearly valuable historic specimens. Studying them gives us an idea of the bird's physiology and its anatomy, what it ate and how it ate it, how it grew and developed. And therefore we can look to see similarities between the extinct great auk and currently living seabirds like the razorbill, which is basically a great auk in miniature. There are controversial projects that are suggested about bringing back extinct biodiversity, and if that was ever the case, samples such as these, which can potentially contain relevant and useful information on what the bird was all about, what it looked like, how it behaved, how it regulated itself, might help with such potential restoration projects into the future. Museums also contain examples of incredibly rare living species. This again is from the Copenhagen Natural History Museum in Denmark, and this is a Sumatran rhino, one of the world's rarest land animals. This Sumatran rhino used to live at the Copenhagen Zoo, but after its death it was taxidermied and is now preserved in the Natural History Museum. Because Sumatran rhinos are so incredibly scarce, studying them can be difficult. So museum specimens such as this can tell us a lot about structure, anatomy, form and function. We might also be able to take genetic information from the tissue samples of the animal to look at, for example, evolutionary pathways with other rhino species, the evolutionary traits that the animal possesses, and how we might be able to look at why the animal breeds in the manner that it does, its physiology, adaptive traits, and any other important information that might help with future conservation action. Famous examples of extinction, such as the passenger pigeon, have taught us that humans can have a long-term impact on biodiversity, which is potentially irreversible. The passenger pigeon is an interesting species to talk about. For a pigeon, it was sexually dimorphic, which is unusual. There is a male in the top of this photo, a female in the middle, and a juvenile at the bottom. The passenger pigeon had these long wings, which it used to help it on its migration. And in fact, part of this bird's scientific name has the word migration in it, to give you an idea of how far it travelled and the distances that it covered. Passenger pigeons used to be common across the North American continent, but human action, hunting pressures, destruction of their habitat, and introduced species that brought novel diseases with them caused the bird's eventual demise, and the very last passenger pigeon, a bird called Martha, died in the Cincinnati Zoo in 1914. The passenger pigeon is a real warning cry from history. It was one of the world's commonest birds, but it's now extinct. If we can understand why these group living animals need to live in large groups to breed successfully, we can work out how to conserve those large groups of species that are still around. We can understand their habitat needs. We can look to see how they can be best conserved, and we can prevent future examples of the passenger pigeon occurring. And that means that many museum specimens that tell us about how animals reproduce are really essential to conservation work. These are the nests of ovenbirds, a South American species that builds its nests out of clay, which then sets rock hard. So called the ovenbird because the clay nest resembles the ovens that local people make to bake their bread. Such artifacts in museums give us an idea of where these particular behaviours come from. And for extinct species, if we have the remnants of a nest or perhaps eggs, we have a whole behaviour encapsulated in time. We've almost got the ghost of the animal's actions left behind, and I find that really interesting. The nest of an extinct bird tells us a lot about the behaviours it once performed. Those types of artefact give us an idea of the importance of the resources the animal accesses in its habitat. So for species like the ovenbird that are still around, how do different populations, different individuals in different areas use resources differently to build their nests? We can track where the animals occur across geography, see how there might be different traits, cultures even for some species in different parts of the world, and we can ensure that those population-specific features are conserved into the future. Many museum specimens are used by scientists to describe exactly what that species is, i.e. they are important for documenting any future finds that relate to the species that is already in the museum. Here's an example from the Field Museum in Chicago in the United States. The photograph of these really woolly mammals are of muskox, a large ungulate that occurs in the Arctic Circle. And this description of the muskox at the Field Museum explains the importance of the specimens that it holds. These muskox are holotypes, and that means the single physical reference which is the example that is used when that species was first scientifically described. This is what a muskox looks like. These are the features that it has. If the environment changes, if the animal adapts with that environmental change, we can look to see how those changes manifest in the free living individuals compared to the original specimens that were collected from the museums. So therefore the museum specimen can document how animals might adapt and evolve to climatic change. We can compare the individuals we have now with the individuals that were collected in the past. The example of the muskox also mentioned lighting and the fact that museum specimens have to be stored under very specific conditions to ensure they're preserved for many years into the future. In fact, in many of the European natural history museums, specimens within them can be many hundreds of years old. So it's important that they are well cared for if they are going to be of scientific value. Lighting is particularly important because individuals kept under bright lights or inappropriate lighting are going to fade very quickly. And it's important that we keep these specimens as biologically accurate as possible if they are going to inform any conservation needs of actually living individuals of that species in the future. This stuffed specimen is of a torrent duck, a South American species that lives in fast-flowing mountainous streams. Torrent ducks are unusual to see in captivity, but they are potentially of conservation concern. So again, what information can be gleaned from the museum specimen? Size, shape, colour, anatomy and physiology, potentially behavioural adaptations. Studying the museum specimens of the torrent duck to find out how they have evolved for a particular habitat could enable us to gather information that's really important to enclosure design or management regimes for living torrent ducks if they were ever brought into captivity in large numbers for any form of conservation action. Of course, for species like the passenger pigeon, the chance for successful conservation action has passed. Unless genomic work, such as the sequencing of the genome of the passenger pigeon, which has occurred, enables this species to be recreated in some manner in the future. These methods that I've already alluded to are controversial, but potential. However, as I've also alluded, studying such museum specimens and looking at their ecology helps us to understand the needs of similar species with similar behaviour patterns. We know that the passenger pigeon had to breed in groups, and we know that these large flocks were really important to its life history strategy. So learning lessons from the past can allow us to ensure that species with these similar traits can be conserved into the future. If we look at some museum examples of species that are dangerously headed for a similar fate of the passenger pigeon, such as these wonderful black rhinos, which are under intensive pressure due to poaching, and in fact, one subspecies of black rhino has gone extinct relatively recently due to the threat from illegal poaching. We should really think about what museum specimens tell us about the form and function and behaviour of these individuals. Look at the size and shape of the horns of these black rhinos preserved in the American Museum of Natural History in New York. The rhino's horn is used for defence and is also a sexually selected signal. So we need to be aware of the function of that particular trait that the animal possesses, and therefore we need to find a way of conserving these populations, of keeping them safe, so these messages that are really important for communication between individuals can be conserved and they can continue to evolve in the future. Likewise, the white rhino, again specimens at the American Museum of Natural History. Huge length of the horns on these old specimens collected in the early 20th century. We need to understand why such traits have developed, the function that they possess, and the reasons behind their display. They are most likely integral to the future success of that individual breeding successfully in the wild, just the same as the large groups of passenger pigeons were so important to the future survival of that species. What traits are possessed by these threatened species that might be further reduced in their messaging and usefulness to that population if that overall population size is further and further decreased by unwanted human action? On a brighter note, museum specimens can tell us a lot about how evolution has shaped some of the weird and wonderful structures that animals possess. Here is a museum specimen of a hornbill, and you can see that the front of the casque, which is the large protrusion that many hornbills grow on the top of their bill, has been opened up, so you can see that honeycomb bone structure inside. Lightweight for flight, plenty of blood vessels to get in for growth and development. Lots of information on the evolutionary processes that have caused the casque to form. We know once again that many species of hornbill are threatened by human activity. The helmeted hornbill, for example, from Southeast Asia, is a critically endangered species, illegally poached for what is termed hornbill ivory. Understanding the features and the function of the casque in a similar way to how we understand the rhino's horn enables us to further direct conservation action by telling us more about species behavioural ecology and why those behaviours have evolved alongside the anatomy of the individual species. And this is particularly important when we look at subspecies and how subspecies within a species complex might be incredibly similar to each other. This is a giant sable antelope, a specimen in the Field Museum in Chicago, North America. The giant sable antelope is the largest subspecies of sable antelope, and it's critically endangered, extirpated from many parts of its range due to civil war in West Africa. Study of museum specimens of the giant sable antelope has shown them to have a very specific evolutionary pathway, and in fact, looking at genomic differences between the giant sable antelope and the other 3 sable antelope subspecies showed that this one split first before all of the others evolved. So there's clearly a lot of ancestral DNA encapsulated in this subspecies, which is clearly worthy of conservation action. So here we can see a real tangible link between the museum specimens and the wild world conservation of the subspecies. But we must remember to look at how well that museum specimen has been conserved and how well that museum specimen resembles the free-living wild individual. If we're going to take measurements of that species, if we want to understand colour, size, shape and behaviour, the museum specimen must best resemble those animals out in the wild. And sometimes that's not always the case. Here we can see a specimen of a king eider with a photograph of an actual living bird. And yes, there is some resemblance, but you can also see many differences too. So it's incredibly important that we check on the quality of the specimens that we wish to study. We have a record of where that specimen has come from, who did the conservation work to preserve that specimen, and how accurate are the measurements going to be from that specimen based on what we understand of the animal whilst it's alive. But what museum specimens do allow us to do very nicely is to look at both their internal and external features close up and in detail to again provide us with information on evolutionary pathways and adaptive traits. So here we've got the external features of the giraffe: pelage, that's its fur, coat colour, ossicones on its head. And then next to it we've got the skeleton, and we can see how the evolution of giraffe is both beautifully shaping its internal anatomy and its external features, the phenotype that we can see. And we can again use that for conservation work by looking at different subspecies, seeing whether or not those subspecies might be full species in their own right, based on DNA samples that tell us when giraffe, as a group altogether, last interbred. And the longer the distance between those interbreeding events, the more likely that the different subspecies of giraffe are actually species in their own right. And if we're also looking at zoo specimens that have come into museums, we can see where those specimens came from originally, how they've been kept in captivity, and what the effect of captivity might be on the size and shape, form and function of the individuals. i.e., do animals change phenotypically? Is there any artificial selection, potential domestication to captivity, which can then be preserved in these museum specimens? And we can look across time to see the long-term effects of captivity on the animal's evolutionary traits. Museums also contain lots of artwork, which, from a cultural perspective, tells us a lot about the human-animal relationship and how we value and document the natural world. Biologically, this artwork is also really important because it tells us what species should look like, how they should behave, and where we find them. As in the case of this painting of these two hornbills, the artist has beautifully captured the posture of the birds, the pair bond between the male and female, and the habitat that they come from. With such specimens where we have large areas of bare skin and tissue, for example on the bird's face and neck, the bill and the casque, those tissues are going to fade in colour very quickly after death as they dry. And during the preservation process, that's also potentially going to change the colour of these tissues. So by painting those particular bits of the animal, based on documented evidence such as this, the museum specimen can be preserved with the correct colours that are straight from the wild. So this artwork is really useful at informing what species display externally, which might drive other important behaviours. For example, anything that's particularly sexually selected. And of course, for extinct species, this artwork is another window onto the past. Here's an example, again from the Field Museum in Chicago, of the ivory-billed woodpecker. Some authorities still believe that the ivory-billed woodpecker exists in the southern United States. Its appearance has not been authenticated since the 1930s. But what we have in this photograph is artwork from John James Audubon, that famous painter of the book The Birds of America, alongside of a stuffed specimen. And we can see that the specimen beautifully matches the artwork. So any preservation of this specimen, what it looks like, how it needs to be positioned, can be done based on the documents of the paintings. And this information again is important for conservation, because any samples that might be found in the wild or any specimens that might be observed can then be compared back to the actual museum specimen and to the artwork to authenticate any potential samples or sightings that might occur in the wild. Documentation, such as artwork, can be really useful in helping museums display their animals in a naturalistic setting. And many natural history museums use dioramas, recreations of populations and communities of species within a naturalistic setting to show off key ecological relationships. Here is an African savanna diorama from the Field Museum in Chicago, and we can see various evolutionary traits that are explained by that species' position or niche within the environment. The grazing mouths of the zebras, the long necks for browsing in the giraffe, and the wallowing behaviour of the rhino. This provides not only an explanation of the form and function of the animals themselves, but also encapsulates the community relationships that occur between species. Should one of these species vanish in the wild, should it only exist in captivity, and perhaps it was bred successfully in captivity for eventual reintroduction, explanations of the community that the species would have come from and the ecological niche that it fits within would be really useful and vitally important information to conservation work. Putting that species back into the right sort of habitat with the right mix of other species is more likely going to lead to successful conservation action and the viability of that population into the future. And of course, when we have very specialised species that have a really specific niche that they fit into, like the shoebill stork you can see in this African waterbird diorama, we can again put the species in the context of all of the features of its habitat. The shoebill has these large toes that spread its weight when it's walking on soft ground. It has this huge heavy bill that it uses for catching lungfish, which occur in these still, slow-moving, stagnant waters of the swamps and reed beds that it occurs in. And the shoebill is not found in large numbers in its habitat. It requires a relatively large home range to live in. Again, this is all explained within this diorama. So should the shoebill be lost from various areas of where it currently lives, we've got that information preserved within the museums, that ecological understanding of how the species fits within its ecosystem that we could use in the future if needed. As well as ecological relationships, museums can also be used to tell us about the evolutionary pressures that drive differences between species within a related group. In this photograph, we've got examples of the pheasant family, displays from two different museums. And you can see in the individuals that are males of their species, they've got colourful plumes and long drooping tail feathers. And they use those tail feathers as part of their courtship display. The males show off, display to the females, and choose their partners. Regardless of the species of pheasant, we've got this similar strategy of displaying colour and feather size and shape to best attract a partner. So by looking across all of these species and seeing the similarities in these traits, even though there are differences between what they look like, we can see how sexual selection and the mechanisms of evolution are acting in a very similar way across all individual species within one taxonomic group. Here's another famous example of sexual selection in action. These are specimens of bird of paradise. To the untrained eye, these all might look very, very similar, but actually they're different species. It just shows how conserved the features of having these drooping plumes and bright colours are between species. This is clearly the best thing to possess to do the most elaborate and most attractive courtship display. When Alfred Russel Wallace, one of the scientists who was working at the same time as Charles Darwin, was exploring the Malay archipelago, he noted that many of the specimens of bird of paradise that he collected contained no legs, and it was told to him that they had no legs because they lived high in the heavens, floating around, drinking on the moisture from clouds. Hence the name bird of paradise. We now know that that's a fallacy, that the bird does have legs, it can perch, and is actually basically a crow in fancy dress. The bird of paradise was prized by the tribespeople of New Guinea and the Malay archipelagos for the beautiful plumes that the bird uses for its courtship display. The legs were simply cut off the specimens because they were less valuable. But that shows the importance of the quality of the specimen within the museum to our understanding of its behaviour and its form and function. We must make sure that the specimen is an accurate representation of what the animal would be whilst alive, so we can see how it fits into its habitat and why it possesses the particular features, so we can understand what they're for. When we have incredibly similar features between animals, we can look very closely at what makes a species a species. Here are 3 examples of cob, an antelope from Africa. They all, like the bird of paradise, look very similar, but the subtle differences between them are related to geography, to habitat, and to location. Species can be quite a difficult thing to define, and in many populations, hybrid zones that naturally occur between subspecies blur the lines of what a species is even more. And again, from a conservation perspective, this is where museum specimens are useful. What should an individual of that species or subspecies look like? What features should or should it not possess? And therefore, with a specimen that has been observed that is unusual, is it a new species? Is it a new subspecies? Or is it a hybrid? The museum specimen can provide all of that information. And type specimens, the original animal that was first collected, to which all others of that species are then described from, are really, really useful at sorting out new events of speciation or simply the occurrence of hybrids. From a captive perspective and a conservation breeding perspective, hybrids are something that's very important to consider, because some species of animal, like the mallard duck and the wood duck, hybridize with other species very, very quickly and very, very frequently. And in fact, here is an example of a hybrid of a wood duck and a mallard. The wood duck from North America is closely related to the mandarin duck from Asia. They're both in the same genus, AIX, A-I-X, but they don't hybridize. So it's interesting when we look at these species relationships of species that should hybridize because they look incredibly similar and they seem to be closely related, actually have some form of biological barrier stopping that hybridization from occurring. So the importance of what hybrids look like, documentation of why they might occur, can again be information gathered from museum specimens. Should any occurrences of this form of hybridization occur in endangered species of populations that are being managed, those hybrids can be identified and it ensures that that doesn't happen again. Conservation is all about ensuring that populations of individuals can produce viable offspring that have all of that adaptive potential to cope with future environmental challenge. The specimens within museums have a value to conservation action by explaining where species are found, by explaining why species have particular traits, why they perform particular behaviours, what they look like, how big or small they are, what their mass is, and what they might look like at different stages of their lives. For species that could be hard to study in the wild, such as the bird of paradise, museum specimens allow us to get up close with many aspects of their form and function which could otherwise be difficult to study. But we must remember that museum specimens are preserved. They are not conserved because they're not alive. And what we must make sure we do with conservation programs is that we don't do preservation. We don't keep our living animals in aspic, as it were. We ensure our living animals are exposed to all of those environmental pressures that would naturally allow evolution to occur. So even though preservation in the museum is final, it can still allow us to do active conservation work by explaining lots of biological information about animals that we do need to conserve into the future, because that information might otherwise not be readily available. I hope you have enjoyed this virtual tour of some of the world's natural history specimens, and I hope it's explained to you why museum specimens, even though they're dead, even though they're no longer breathing, can be useful to conservation science. The more we study the specimens we have in museums, the more we can understand about the biology and ecology of those animals that are preserved there. More can be learnt about past mistakes to stop them from happening again in the future. Thank you very much.